Right, so we'll go right on to our next speakers. Our second podium presentation is going to be on research conducted by School of Nursing and School of Medicine team that includes doctoral candidates and faculty. So we have Brittany Knight, Jessica Yasko, Stephen Crocker, Aaron Young, and Kyle Bombauer. And their presentation is titled, as you can see, The Role of TIMP1 in the Development of Hypersensitivity Using a Model of Cutaneous Inflammation. And they're seeking to understand underlying mechanisms as well associated with hypersensitivity, which is a significant contributor to chronic pain. So please help me welcome, is it Brittany that's going to be speaking? Yes.
Just need some water. So hello, I'm coming from Farmington today. I'm a doctoral candidate in the Biomedical Sciences program at UConn Health in Dr. Kyle Bombauer's lab. And as you mentioned, I'm here just to share a little bit of my data concerning my thesis looking at the role of TIMP1 in the development of hypersensitivity using a model of inflammation. So pain is typically characterized as being acute, which is indicative of the protective phase. When you have a minor wound or injury, your tissue needs to heal and then regenerate the lost tissue that may have been injured during that wound. And so it's typically a shorter period of time in humans, less than 30 days. And if you're uncomfortable, you can take an anti-inflammatory drug, and that usually is pretty effective at treating those symptoms. However, the underlying issue becomes a significant medical problem if that pain persists past that time period of repair. And it can last for months or even years in duration. And unfortunately, this is seen and argued in the field as being chronic pain being a disease in itself. And it'd be very hard to treat. There's no cure available. And so I'm talking about today's theme in vulnerability. Poorly treated pain can lead to functional impairment in any group of people that are dealing with symptoms of pain. However, in talking about populations that are vulnerable, the elderly can actually become socially isolated, which can lead to the development of depression or a cognitive decline. Another population that can be targeted are the individuals in lower socioeconomic status. The financial burden is significant with dealing with chronic pain over a period of time. And if there's no cure available, these individuals can be financially stressed into dealing or managing with their symptoms, which can actually exacerbate their symptoms of pain. And it's also important to know that when we're talking about pain, although the individual is the one experiencing it and the physician or another clinician is helping them to manage their symptoms, the pain management in general is actually bigger than that. It involves the FDA, the pharmaceutical companies, insurance companies, getting access to medical devices that may not be able to be given after you've had surgery or had your disease over a period of time. And so over to the right, I just have some statistics on the prevalence of two chronic inflammatory diseases, rheumatoid arthritis and psoriatic arthritis, their prevalence in the United States and the world, as well as a mean average cost per person that it takes to manage these symptoms. So when talking about how we can try to better provide therapies for individuals who have chronic pain, inflammation is known to be a biological risk factor for developing chronic pain. And initially when you have an infection, an injury, or just cell damage in general, your body's first response is to eliminate that cause of cell death and to initiate repair. And there are 5 symptoms typically associated with inflammation: redness, immobility, swelling, heat, and pain. And as I mentioned, the acute phase of that, as illustrated at the top right, is that there's an acute inflammatory response that once the repair has happened, usually your symptoms of pain subside. And there are molecules that are released in your body to help tell your inflammatory system to stop. We're good. Everything's been fixed. However, sometimes that cue can be lost or muffled if there is a significant more amount of inflammation. That can lead to chronic inflammation, which is significant because although inflammation is a secondary component of injury, it also can be a precursor to the development of other diseases such as cancer, arthritis, autoimmune diseases. And so we know acutely that targeting the inflammatory process is pretty effective at attenuating pain through the use of steroids or non-steroidal anti-inflammatory drugs such as ibuprofen. However, identifying molecules that are important for this transition from acute to chronic pain are what we were really trying to figure out. How does our body know when to turn off the pain signal? And why does that not happen in chronic conditions? So that's not supposed to show up yet. Okay. I have some transitions, but I guess they're not working.
Your actual slideshow.
Okay. Oh, okay. Sorry about that. Okay, no problem. So some molecules that have been identified to contribute to the development of chronic pain, such as in following nerve damage, neuropathic pain, are these molecules called MMPs, or matrix metalloproteases. And these are enzymes that are important for the wound healing process when you have an injury. They help tear up the protein scaffold your cells need to sit on so they can function and then return back to homeostasis. And so in this paper, not to get too much in the basic science, they used a nerve injury model, and they showed that, just to orient you, so this is a behavior graph, and we'll see a few more of these. The y-axis is in Paul withdrawal threshold, so how much force is needed to elicit a mechanical response. So we're looking at mechanical sensitivity, which is a phenotype or a symptom of chronic pain, so mechanical sensitivity. And they show that compared to baseline, if we look at the bottom line, that's the spinal nerve damage group, that they have an increase in sensitivity or drop in threshold as soon as 1 day following nerve damage, and that is persists for about 2 weeks. If they administer a small molecule following nerve damage, they can actually attenuate that sensitivity by inhibiting MMP specifically, and that's shown in the top line here. So they become, there's an attenuation of their pain. Interesting to me in the lab, so they use a small molecule inhibitor to prevent or inhibit MMP9, but the endogenous inhibitor that our body produces to inhibit MMP9 is TIMP1, tissue inhibitor of matrix metalloprotease 1. And in the B panel, if you just look at the solid white bars, that is the nerve damage group, so there's that drop in threshold or increased sensitivity following nerve damage, and then it persists for a period of time. But in animals that receive a TIMP1, as opposed to the MMP9 inhibitor, there's an attenuation in that pain phenotype, which suggests that TIMP1 alone can produce an analgesic response following nerve damage to reduce pain. And so I just told you that inflammation is a driving component of pain or chronic pain, and although they use one model, nerve damage, other individuals can have develop persistent inflammatory pain in the absence of nerve damage. So I wanted to ask the question, how does TIMP1 contribute to the development of inflammatory pain? Can we harness its potential analgesic function to then prevent persistent inflammatory pain? And so in the lab, the way we do this is we use mice, these black 6 mice, and I administered a form of sterile inflammation, complete Freund's adjuvant, or CFA, which is heat inactivated TB. So there's no bug driving that infection, it's sterile. And I subcutaneously injected into the right hind paw of mice. And then to understand whether or not TIMP1 contributes to this development of sensitivity, I wanted to first see if inflammation can cause or change TIMP1's expression in skin. And so I collected tissue from mice at 1, 3, 5, and 7 days. And in this graph, I'm showing you total TIMP1 expression in skin from these mice using an ELISA, or an enzyme-linked immunoabsorbent assay. And so compared to naive controls, at 1 day there's a significant upregulation of TIMP1 in skin, and it stays elevated for about 7 days. And so to identify in skin, where is this TIMP1 even coming from? We looked at one cell type that's been shown to contribute to the pain process itself. Although neurons are important for that process to send to the brain for the cognitive component of pain, non-neuronal cells in skin can also contribute to this process. And so we looked at keratinocytes marked with K14 here. This is immunohistochemistry. So we're looking at an image of skin that I use different markers to identify different cell types, such as keratinocytes and then TIMP1. And so on the top part of this graph is the naive condition, and consistent with our ELISA data, we see that there's a very low basal level of TIMP1 in skin. But immediately following CFA, there's a significant upregulation, which led us to believe that perhaps following inflammation, keratinocytes are responsible for upregulating TIMP1 in skin. So that's great. And how can we correlate the expression of TIMP1 to the development of sensitivity following inflammation? And so to do this, I used that test that was similar in that paper I showed you a few slides ago, looking at mechanical sensitivity. And so this is change from baseline. So I normalized all animals to their baseline sensitivity. And on the y-axis is sensitivity. So going down, following the arrow, is a reduction in threshold or increase in sensitivity. And so in my ELISA data, I showed you that if one day following CFA, there's a significant increase in TIMP1 in skin. And we don't see a change from baseline at 1 day. We see the increase in sensitivity at 3 days. We think that robust increase of TIMP1 at 1 day compared to naive may be important for preventing the onset of inflammatory sensitivity in wild-type mice. So to test that, this is my question. So how does TIMP1 regulate the development of sensitivity following inflammation? We decided to compare our wild-type mice to a global knockout for TIMP1. So a TIMP1 knockout, also referred to as ATHENA. And so I told you that we're interested in correlating the expression of TIMP1 to the development of symptoms associated with chronic pain. And one of those symptoms is the increased sensitivity in non-inflamed tissues, the tissues that aren't damaged. And this can happen in a variety of chronic conditions. And so to test this in the lab, we subcutaneously injected CFA into the top of the hind paw and then measured sensitivity on the bottom paw, part of the paw, which is not inflamed. And we used, again, these bone-free filaments to measure mechanical sensitivity. And when we do this, to orient yourself, all the dotted lines are the naive controls. We didn't see any differences in baseline sensitivity between genotypes. And then the pink line is the wild type inflamed. So I just showed you this. They become sensitive around 3 days, and then it's resolved by 7. However, in our TIMP1 knockouts, there is a significant reduction in threshold or increased sensitivity at 1 day, but then persists for 7, which suggests that TIMP1's expression early on in the inflammatory process may be important for allowing pain to resolve in these wild-type mice, which is lost in our knockouts. So the next thing we wanted to ask is if we can put this back into knockouts, the TIMP1 knockouts, can we prevent their development of sensitivity following inflammation? If this is the molecule that is setting up the system to resolve ultimately. And so to do this, again, we used our TIMP1 knockout, and we administered CFA, our inflammatory stimulus, and then recombinant TIMP1. So we're putting it back in the animal, and again, measuring mechanical sensitivity for 7 days. And previously I had conducted a dose response curve, and we found that there was an effective dose of about 10 nanograms per microliter at the time of CFA injection in these knockouts. So again, to orient yourself, the dotted line is the naive group. The pink line is the CFA alone group, which I just showed you on the previous slide. And when these knockouts, awesomely enough, when they were administered TIMP1 at the time of their inflammatory stim, their sensitivity at 1 day was lost, which suggested that TIMP1 is very important during that onset of inflammation to then help prevent that hypersensitivity and allow pain to resolve over time. So in conclusion, I showed you today that cutaneous inflammation can upregulate TIMP1 expression, and we think that that is occurring in keratinocytes, and that release from keratinocytes is affecting the pain circuits. The upregulation of TIMP1 expression in skin seems to occur before the onset of mechanical sensitivity in wild type, so we think that this is a temporal is important at that onset of inflammation. And that we saw that in wild types, being that TIMP1 was expressed already that it was able to prevent their onset of inflammatory sensitivity, and that was lost in knockouts. And that the replacement of TIMP1 attenuated sensitivity in our TIMP1 knockouts, that they were able to resolve their pain and they became less hypersensitive. In big picture, we think that the administration of TIMP1 may serve as an alternative method for preventing or treating persistent inflammatory pain. It might be that molecule that is important for your body's cue to know when to turn off and resolve pain following that initial wound. So to acknowledge my lab at UConn Health, Dr. Kyle Baumgauer, Jessica Yaskill, I had the pleasure of working with several undergraduate students in the honors program, Nate and Layla and Sarah, and then Justin from UHeart, as well as collaboration with Erin Young and her student Noel and their tech Nikki, my committee members at UConn Health. Also extend a thank you to the biomedical science program, the School of Nursing, of course, for all their support, and UConn as a whole. Thank you. Yes.
So it sounds like in the last part of the experiment, you gave the mice TIMP1 at the same time that you stimulated the inflammation.
Yes.
But then your last statement was, well, maybe TIMP1 can help people with persistent inflammatory issues resolve that. That seems like a big leap, because the beginning insult is, for many people, is years before they come to a doctor or nurse about pain.
For sure. Yes, so good point. It's known and it's been shown in that nerve injury model that TIMP1 is important for that onset and that development, and that another TIMP is actually important for that maintenance. I'm simply proposing that maybe if individuals that may be more at risk for developing that persistence of inflammation, that TIMP1 may be important for preventing that or attenuating that, maybe following nerve damage or any other conditions that they may have. But you're right. I don't know if it would be helpful later. I haven't done that experiment to see if it would resolve that pain. Any other questions?
Am I allowed to ask a question? I'll be nice.
Very nice presentation. Of course, expected. So what do you think is important about TIMP1 instead of just strictly going in and inhibiting the MMPs themselves? So clinically that's been done. Why do you think TIMP1 is going to be the key to the success of solving this problem of potentially persistent inflammatory pain?
Yes. So, as you mentioned, TIMP1 is primarily known to inhibit MMPs, and in that neuropathic pain model, they showed that presumably TIMP1 is inhibiting MMPs to prevent the persistence of neuropathic pain. However, in my hands, I'm not necessarily seeing that. I think TIMP1 is actually a very multifunctional protein. It is also can bind to a cell surface receptor that can facilitate gene expression that is involved in pain, and I think that it has a signaling peptide that then can affect the system, and I think that's what's driving this analgesic effect. So it's very exciting because most of the field is just worried about TIMPs and MMPs, and I'm going to show a very novel pathway that I think may be important for this pain processing and its potential as being an analgesic molecule.
Any other questions? All right, then, do you want to help